At one point in the 20th century, memory research got so strange that scientists trained flatworms, ground them up, and fed them to other flatworms. The idea was that if learning changed the body, maybe some part of that change could survive in the tissue. Maybe the second animal would inherit the memory of the first. It is the kind of experiment that sounds ridiculous until you realize they were asking a serious question. If memory is real, where does it actually live? For most of us now, the answer feels more obvious, but the history of memory science is not that clean. The question of where memory is stored has been haunting thinkers for a very long time. One of the oldest images was memory as an impression, something left behind by experience. Thousands of years ago, Plato described memory in theateus. Here, the mind or soul was imagined almost like a wax tablet, where perceptions and thoughts could be stamped into it. If the wax was smooth and deep enough, the impression could last. If it was too soft, too hard, or disturbed, the impression could blur. Of course, that is not how we see it today, but the metaphor still resonates. Memory feels like something has been impressed into us. Something happened, and after there is a change. Later, scientists began searching for that impression more literally. In 1904, a German biologist named Richard Semon gave the memory trace a name. He called it the engram, the thing that made it possible for a past event to be stored, carried forward, and later reactivated. Semon's broader ideas did not survive, but the term did. Then in the 1920s, one of the most famous searches for where memory is stored came from Karl Lashley. To test this, he trained animals, especially rats, on tasks like mazes. Once trained, he would surgically remove or destroy different parts of the cortex to determine whether the learned behavior disappeared or not. Essentially, he wanted to locate the precise place where memory lived, the drawer, the file, the spot in the brain where it was stored. But that is not what he found. The more cortex he removed, the more performance could suffer, but there was no single spot where the memory of the training seemed to live. Instead, Lashley's work suggested that memory is more distributed, spread out, less like a file in one drawer, more like a pattern across a system. By 1949, Donald Hebb helped shape the question again. Maybe memory was not stored in one location, but in the changing relationships between neurons. Neurons that activate together can become more strongly linked. Connections can strengthen, circuits can change, and this became one of the most popular ideas in neuroscience. In this view, memory was not one substance in one place. It was a change in a network, a pattern of altered connection. That idea is still carried forward today. But in my research, I stumbled across another take. In 1953, Watson and Crick published the structure of DNA, and biology started changing. After this, life was seen through molecules that carried information, codes, messages, instructions, and once inherited information could be stored in chemical form, a question became inevitable. Could acquired information leave a molecular trace too? Not like a whole movie playing in the mind, but some biological residue of experience, some molecule or molecular pattern that helped a memory persist. And this is where the history starts to bend towards the strange, because for a brief moment in the 20th century, the idea of a memory molecule did not sound like madness. It sounded like a possibility. And one scientist followed that possibility right into the flatworm. Now, admittedly, this does not look like an animal that should make anyone think memory. It is tiny, soft-bodied, simple-looking, almost like a little brown ribbon moving through water. But a type of flatworm called a planarian can do something extraordinary. They can regenerate. If a planarian is cut in half, the head can regrow a tail, and the tail can regrow a head. Not just a scar, not just a repair, but a new head with a rebuilt nervous system and a simple brain-like structure. And that is where its role in the memory question became obvious. If an animal can regrow the part of its body that contains its quote-unquote brain, then what happens to something it learned before the brain is rebuilt? The scientist who became most associated with this question was James V. McConnell, a psychologist at the University of Michigan. And before his work became infamous, the first question was surprisingly straightforward. Could a planarian learn? In the 1950s, McConnell and Robert Thompson tested whether planarians could be classically conditioned. They paired light with a mild electric shock. At first, the shock produced the reaction. The worm would contract. But after repeated pairings, they wondered whether light alone could begin to trigger that response. In other words, could this tiny animal form an association? Could it remember that light predicted shock? McConnell and Thompson reported that it could. And once that possibility was on the table, the next question became even stranger. If a planarian could learn, what would happen to that memory during regeneration? To test this, McConnell and his students trained planarians, then divided them into head and tail sections. Separately, the head and tail portions regenerated what it had lost. Then they tested the regenerated animals again. And according to McConnell's early reports, the regenerated worms appeared to show signs of retaining the training. Even the tail pieces, which had to regrow a new head, seemed to relearn the response faster than completely untrained worms. This result changed the trajectory of McConnell's research. Some part of the training seemed to have survived the rebuilding of the nervous system. And for McConnell, that opened a door that would take the research somewhere much wilder than regeneration. He thought if memory could survive this process, then maybe it was not only a pattern in the original brain structure, maybe some part of it was being carried by the body itself. And in the molecular excitement of the 1950s and 60s, one possible answer began to look tempting. Maybe memory had a molecule. To test this, McConnell and his group started with direct approaches. They attempted to graft tissue. They tried injections. But planarians are tiny, soft animals, and the technical problems made this extremely difficult. Then came the method that made the experiment famous. Knowing that some planarians will consume other planarians, McConnell's group decided to train the worms, grind them up, and feed that to the untrained worms. Essentially, they wondered, could trained tissue make an untrained animal more likely to acquire the same response later? According to McConnell, the answer appeared to be yes. The recipient worms did not instantly behave as if they had been trained, but they reportedly learned the response faster than worms that had not received material from trained animals. To McConnell, that suggested that something like a memory molecule from the trained worms had survived and somehow influenced the behavior of the untrained worms. And that was the claim that made the story explode. Because once you say trained tissue may affect memory related behavior in another animal, the careful scientific question almost disappears. What people heard was something about memory moving through matter. Quickly, the experiment became famous, and McConnell leaned into the attention. The work entered magazines, television, and popular science. People talked about memory pills and memory injections. But the more dramatic the story became, the more pressure and evidence it had to carry, because a claim like this cannot survive on one result. And for a while, it became a real research wave. If memory could really be influenced by matter from another animal, other labs wanted to see it too. The results needed to be reproduced. Labs tried versions of this experiment in planarians, rats, goldfish, honeybees, octopuses, baby chicks, and other animals. Some studies reported positive results. Some seemed to support parts of the idea, but many did not. And the more that people tested it, the harder the pattern became to trust. The results were inconsistent. Different labs got different outcomes. And with a claim this strange, inconsistency quickly becomes a serious problem. It didn't take long before researchers began to have more objections. Were the animals really receiving something memory related? Or were they simply being changed by stress, sensitization, nutrition, or some other biological effect? And then there was the problem of measurement. Planarians are not easy subjects. They are tiny, their behavior is subtle, Training and testing could take hours, and much of the early work depended on human observers watching small movements and deciding what counted as a response. And that does not mean the observers were dishonest. It means the experiment was vulnerable. When effect is small, strange, and hard to measure, bias can enter quietly. And suddenly, two labs can believe they are doing the same experiment while actually testing slightly different things. McConnell's public image complicated things too. Sure, he was charismatic, he was funny, he liked the publicity, but he talked about memory pills and memory injections. And then there was his Worm Runner's Digest, which was mixed with real scientific work, satire, jokes, cartoons, and deliberately absurd pieces. All of this together made it easier for critics to see the whole thing as unserious. So the field became tangled and eventually scientists moved on. Memory transfer did not become accepted science. RNA did not become the simple storage molecule for experience. And McConnell's work became a classic cautionary tale for new scientists. But this is the part I find most interesting. The failure of those experiments did not erase the original question. It only made the question harder to ask, because underneath the controversy, there is still something real. Memory has to become biology somehow. Experience has to leave a change somewhere. And once some of the stigma wore off decades later, the question returned. Not in exactly the same form. This time it came back as a more cautious version of itself. In 2013, researchers Tal Shmarat and Michael Levin published a study using a more automated training system to revisit the question of memory after regeneration. Levin's group tried to make the process more objective. They trained planarians in a specific environment associated with food, tracked their behavior with automated methods, then removed their heads and allowed them to regenerate. After the worms grew new heads, the researchers tested whether the earlier experience still mattered. The strongest result came after a brief re-exposure to the same environment where the previously familiarized worms reached the feeding criterion faster than controls. And then in 2018, RNA entered the story again, this time not in Planarians, but in Aplysia, a sea slug that had been used for decades in memory research. The paper was called RNA from Trained Aplysia Can Induce an Epigenetic Engram for Long-Term Sensitization in Untrained Aplysia. Even the title itself made it sound like the old memory molecule question had returned in a modern form. Here they were studying a form of memory called long-term sensitization. In aplysia, repeated tell shocks can produce long-term sensitization, a lasting increase in the animal's defensive withdrawal reflex. After that experience, a light touch can trigger a stronger response than it did before. This is memory in a very specific sense, where experience leaves a lasting change that alters future behavior. Then they extracted RNA from the animal's central ganglia and injected that RNA into untrained aplysia. And according to the paper, the untrained animals that received RNA from trained donors showed an enhanced withdrawal response compared with animals that received RNA from untrained donors. But then the study went further. The researchers also tested neurons in culture. And when sensory neurons from naive animals were exposed to RNA from trained animals, those neurons became more excitable. They found that the effect was also selectively increasing excitability in sensory neurons, but not in motor. And when the researchers blocked DNA methylation, the RNA-induced behavioral effect was blocked. So the authors argued that RNA from trained animals could induce an epigenetic engram for long-term sensitization in untrained aplysia. The paper describes this as support for a non-synaptic epigenetic model of memory storage. That is an impressive claim. The 2018 paper itself even acknowledges the field's history. But this is not a simple comeback story. Decades later, the question returned with better tools, better model systems, more carefully defined methods, and a much more restrained interpretation. In the first iteration of this experiment, the public story became bigger than the data. McConnell's work became a cautionary tale. But underneath all of that was a question that never stopped mattering, even to this day. If memory exists, it has to become something, some record of experience. That does not mean memory is simple. It does not mean memory lives in one place. It may be distributed across circuits, synapses, gene expression, cellular state, and time. And perhaps the deepest versions of this question go even further. Maybe some memories are not stored as objects inside us at all. Maybe they are patterns a living system can retrieve under the right conditions. McConnell's life took strange turns after his work. First, he became famous, then infamous, and oddly enough, that seemed to result in him becoming one of the targets in the Unabomber campaign. He was only injured, his hearing was affected, but what a bizarre and violent footnote in the life of a scientist already tied to one of the strangest stories. The part I applaud is not the spectacle around him. It's the question he could not let go of, the experiment he was not afraid to try. Because even if the answer is wrong, the courage to ask the question can still change what science is willing to test and see in the future. And even though the memory molecule may not be the answer, it certainly left behind its own kind of mark. I cannot wait to hear your thoughts on all of this. And until next time, stay curious and take care.